Welcome to our first HAWK Biosystems webinar. Thank you all for your time and interest in HAWK Biosystems and our technology. And I'm very pleased to introduce Professor Peter Parker, who's going to give a webinar and a presentation on functional biomarkers. I think we'll then have time for a couple of quick questions if there's anything pertinent to Peter's talk. Then we'll move on to a second presentation where I'm James Miles, the product manager of HAWK Biosystems. I will just introduce some comparative data between our QF-Pro technology and other spatial biology technologies. And then we'll move on to a full Q&A session and we can take as many questions and have some scientific discussion, I hope. So, yeah, I'm very happy to introduce Professor Peter Parker. Peter is chairperson of our scientific advisory board and co-founder of HAWK Biosystems. And just a brief introduction on Peter. Peter was awarded a Doctor's Philosophy in Biochemistry from Oxford in 1979. Following postdoctoral fellowships, he became a principal scientist at the Imperial Cancer Research Fund. And Peter's been elected to the European Molecular Biology Organization, the Academy of Medical Sciences, and the Royal Society in 2006. So I'm very pleased to, I think, hand over to Peter, who's going to give a very interesting webinar on the importance of functional biomarkers and how they can transform research. So Peter, over to you.
Thank you very much, James. I hope everyone can hear me clearly. I'm sort of delighted to have this opportunity to climb back on a hobby horse of mine, which has been functional biomarkers, something I've been passionate about for decades now. For completeness, my affiliations are the Francis Crick Institute and King's College London, and I should also include the Medical Research Council that I work for, and then the companies HAWK Biosystems, Apollo Therapeutics, and Foremost. These sorts of diagrams are probably familiar to many of you, and don't worry, I'm not going to dig into any details around this. I just want to discuss a few principles because, of course, the sorts of regulatory relays that control cellular responses, their homeostatic behaviors, their response to stress to the environment, and so on, sit latent within cells. These relays are there, ready to respond as and when necessary. And it's a series of effectively, these are switches, perhaps not just simply on off, but perhaps dimmer switches that allow the cell then to respond appropriately to its context. And it's not the concentration and the presence of these. These are typically present in many, if not all cells in this sort of complex of mixtures. And this is just a very small variety of the types of relays that exist. But it is the state of these that is important. And if you walk into a room, the number of switches in a room doesn't define the ambient lighting. It is the state at which lights are on or off within that room. And it's really this that for me defines what I'm going to talk about, which are functional biomarkers. And it is what is those regulators, what is their in or activation state that is then telling us about the throughput of information to which the cell is responding. And of course, this is critically important, not simply in physiology, but in the pathology of disease. Okay, it's not about the presence of these regulatory relays. It is about their activation state or inactivation state. So it is the state of these. This is what really, for me, defines then functional biomarkers. And to be useful in terms of our being able to monitor these biomarkers, I always felt that we needed to be able to do this in the context of what is a typical a sample, whether that's a cell, a fixed cell, whether it is a sample of tissue from a model system, or indeed from a patient. And of course, the routine path lab sample would be a fixed section that can then be stained up with an antibody to look at whatever it is that is going on. And of course, most of what's done in a pathology context is to look at the concentration of things. And that is absolutely not what we're talking about here. We're talking about what is the state of those things within that sample. And so the states, whether they're in complexes, whether they have been modified in a way that is evidence of them being switched on or switched off, is really where the functional biomarkers come to play. And to be able to do that in the context of maintaining the integrity of that tissue sample was also an important part of how do we then monitor this in a useful way. Of course, nature lends itself to being able to tackle some of these problems because, of course, protein interactions, protein modifications, these are tractable problems with the use of antibodies that can allow us to see then when things are appropriately changing in that domain. However, there are significant issues with that. And just to illustrate that point, one of our original motivators for developing the technology I'm going to describe today was a protein what we knew got modified, and we could raise good antibodies against that modification. But these antibodies were not monospecific. They would recognize our protein, but we knew they recognized at least one other protein. And of course, in an unknown patient sample where we wanted to look at the activation state of this protein, we would not be able to distinguish our protein from any other protein. And so we set about then coming up with a methodology that would allow us to really drive the specificity and came up then with a solution that I will briefly describe. And this is based on force to resonance energy transfer. So the proximity of two fluorophores is such that a donor fluorophore will change its lifetime in the presence of a resonating acceptor fluorophore. On its own, it has a defined lifetime excitation. It'll go into an active state, and then it'll depopulate that state in a stochastic fashion. If it's resonating with another fluorophore, that lifetime is greatly shortened, and we can measure that with great accuracy. And so we can exploit this to simply stoichiometrically label up a pair of antibodies. So one, perhaps in the case that we were interested in, against our very particular protein that was selected and could be very specific, and one against this modification. So whether it's a complex as illustrated here, or whether it's a post-translational modification. Obviously, in the absence, there is no resonance. In the presence, there is, and you can detect that with great accuracy. And this worked brilliantly for us. So we could show that there was a characteristic pattern of activation in the context of certain cancers. Now, this was a great tool, but it was a hard to use tool. You had to be precise about the labeling of all of these components, and you also had to do it for each antibody you wanted to use. And of course, not every antibody pair is going to work in the way you want it to. You have to screen a variety. And that really led us then to look for how can we make this user-friendly and accessible to more problems, more people. And this then led us to generate a more generic solution to that. It's still exploiting fluorescence resonance energy transfer, but using secondary reagents that could detect specifically donor pairs of antibodies, and an amplified system for the acceptor. And this was the basis of the technology then that was developed and licensed by the Francis Crick Institute to HAWK Biosystems, and that's the basis of the QF-Pro platform that James will talk about later. And this works, again, this works just as well. In fact, it works better than the system we'd originally started. But in fact, it has that sort of general usage in that you can very easily now screen different primary antibodies, pair them up and test them using these secondary reagents to look at then the proximity in running these assays for complexes, in running these assays for post-translational modifications. And of course, the specificity this brings is really exquisite. It's like running a two-site ELISA assay. You're really bringing specificity and quantitation in the nature of this readout to the system. And so, just to give you a couple of examples in some of the research we have done using these sorts of tools, I'm sure you're sort of familiar with the fact that, like patients, mice are not particularly compliant when it comes to taking drugs when you're trying to validate pathways in terms of drug interventions. And we have used these sorts of tools to look at the activation state of targets within tumors by taking tumor samples and running immunohistochemistry type studies, but using this FRET-based assay as a readout of activation state to ask the question whether does the drug treatment correlate with changing tumor growth, and is it that those mice that are treated with that drug who do not respond where do they sit on the spectrum of behavior? And actually, the answer is very simple, and as sort of illustrated here, and I put a green circle around it. Those mice that respond to drug in terms of changing that target and deactivating the target, so there's a reduction in tumor growth associated with a reduction in the activation state of that target. It is those mice that is the clear correlation. And there are a few mice who do not respond in terms of a reduced tumor growth. And it turns out that the target is not changing in those mice. And so we can come to a clear correlation that gives us great confidence then in validating that target and then being able to move forward with a drug development program or indeed selecting interventions. Now, this was done with mouse tissue, but of course you can do this with human tissue. And in fact, for exactly that same target, if you look at triple negative breast cancer, it turns out then that, and you can see from these 3 patients that are just simply examples of the sort of information you can get in this pseudo color range on the right hand side, where there is high FRET and high activation state, colored orange, that in patient 1 there is a spectrum, patient 2 less so, patient 3 even less. But the correlation between prognosis and the activation state is such that high activation state is a poor prognosis for these patients. And this bears no relationship, I emphasize this bears no relationship to the concentration of that pathway per se. It's not about the concentration, it's about the activation state. Again, functional biomarkers, I think, are really important in this context. Now, one of the really, I think, innovative things that HAWK did in developing and exploiting this sort of technology was in the development of an assay that would allow you to look at complexes that exist between cells. So the presentation of a ligand on one cell and a receptor on another cell, and developing a method then that exploits this sort of technology to be able to see these. And this really has profound implications in terms of immune oncology. And just to put that into some particular context, so the presentation of a ligand on one cell can be important in suppressing the ability of a T cell that's approaching that cell to respond in terms of any cytotoxic consequences. And so there are lots of so-called immune checkpoints that exist, that are being characterized, and this is one of them. This is the interaction of TIGIT with CD155, where you've got cell-cell interactions that determine the nature of the immune surveillance system. And being able to see that complex in situ is something that has then been developed by HAWK and has been really informative in terms of immune oncology. Now, you're probably more familiar, many of you, with the PD-1, PD-L1 story. There is an immune checkpoint that has really made some inroads into the treatment of cancers, and cancers typically exploit the suppression of the immune system in terms of their own survival, and our ability then to negate that suppression of the immune system has a profound influence in terms of an attack on the tumor. And so in non-small cell lung cancer, the evidence using the QF-Pro platform is that if you look at the complex between PD-1 and PD-L1 as a measure of its to look at its engagement, this is a functional readout then of the engagement of that system in the immune suppression that pertains in non-small cell lung cancer. You get a tremendous separation between those with a high FRET efficiency that reflect a very good response to drug compared to those with a very low FRET efficiency. So in other words, very little complex, very little response to drug. And that drug is simply a drug that blocks the interaction between the two. And in a sense, it's sort of logical that if there is no interaction, you're not really going to have a profound effect on tumor outcome by putting a drug in that influences that interaction. Where there is extensive interaction, it seems as though, in fact, that is important. It is rate limiting in that context, and you are getting a very good response to drug. Now, what's used generally in clinics to date has been the concentration of the ligand. I repeat, the concentration of the ligand, so the PD-L1. If there's a lot of PD-L1 around, patients get treated in first line. But actually, that's a very poor surrogate of what is going on. It really has limited information. On the right-hand side, you can see there is a poor separation between those with a high PD-L1 and those with low score PD-L1. And that's not just the data from this analysis. Others have also noted this. And if you really break this down, and we just look into a little bit of detail around this, what you can see here then are the sort of four groups you get of high complex formation, so high FRET, high ligand, and in the bottom left-hand quadrant, you've got low FRET, so low complex formation and low ligand. And first of all, to say that really where you have high ligand, what's really important is actually not so much the high ligand, it is the complex formation, the high FRET that you see between these. And in fact, in this cohort of patients, and this is some 150 or so patients, within that 4-year window, they've not even reached the 50% overall survival limit. So there is a really profound separation then of responders and non-responders here. And if you look again, if you look again then at those that have high FRET, even with low ligand, there is a very clear distinction, implying then that the low ligand, where normally you wouldn't necessarily get a first-line treatment with an intervention of this type, at low ligand, the high FRET would say that actually this is it's the wrong decision. Really, you should be treating these patients. And so we have these groups then that are low FRET, high ligand, where it's not clear really whether the right decisions are being taken, but particularly then those, I think, where there is benefit to be had, where there is complex formation, but low ligand levels, where treatments really should be deployed. So there is this, this platform brings us very particular opportunities then in driving forward the selection of therapies within the context at least of non-small cell lung cancer, and no doubt other cancers with that all employ ways of which they avoid immune surveillance. But it's a great deal more than that. And firstly, to say that really that platform is the only commercially available platform that gives you this type of information. It is quantitative. It gives you molecular resolution. So the FRET itself operates below 10 nanometers, a single-digit nanometer. So we're really talking about proteins that are touching each other. We're not talking about proteins that happen to be in the same cell or happen to be in cells next to each other. There is clear specificity that's driven by this combination of antibodies that is really important, I think, in giving us confidence that the types of information we are getting back is appropriate, and it is very sensitive in terms of what the level of detection. And really, all of the other systems that are currently around are proximity and associated correlates that really don't get down into the sort of molecular details that I think are demanded by the sort of functional biomarkers that we require. And no doubt James will cover a bit of that later. Now, of course, this isn't just about IO and non-small cell, as I said. There is a lot of opportunity here in terms of not just the clinical domains, but it's really an unequal tool in terms of research and development. And so, yes, there are other IO applications, and we certainly have looked at, and HAWK have looked at melanoma, head and neck cancer, and others. And there are other associated biomarker readouts in oncology. But in terms of immune disorders, in terms of infectious diseases and neurodegeneration, and so on, in a number of other therapeutic areas, there are opportunities to drive functional biomarkers that give us precise readouts associated with regulatory pathways associated with macromolecular interactions, protein-protein, protein modifications, protein DNA, that lend themselves to the types of technology that we are talking about here. So the platform that you can use to read this, these sorts of attributes, the Violet 3 system, so this is third generation, is really a very user-friendly system. It gives quantitative, as I said, this is quantitative and reproducible analysis. You get great specificity and good signal to noise. This is an instrument that's dedicated to these types of readouts. So it's not an adaptation of something else that may or may not quite give you what you need. And it's accompanied by really good software that allows you to run these analysis. And of course, the proprietary secondary reagents are available to allow you then to screen your favorite antibodies and to test what you need to test. So in terms of these sorts of functional biomarkers that where we started, you can do this. You can take tissue samples, whether they are, or cell samples, whether they're from the lab or whether they're from clinic, and you can look in cells, in organized mouse tissues, patient biopsies, at the states of proteins and the states of protein-protein interactions, protein macromolecular interactions, and determine this with some accuracy and precision. And this is really valuable, not only, I think, in that discovery and development and in the clinical, and indeed also in the clinical setting. And it's been incredibly valuable to us. And if there are some specific questions, as James said, happy to do that before we launch off into the wider discussion with James.
Okay, I can't see any hands up. So in which case we'll proceed straight on to the second half of the webinar, where I'll briefly present in a bit more detail what other technologies are operating in this area and how they compare with each other. And then afterwards we can head to a more general Q&A session. Is everyone able to see my screen okay? And critically, is it moving?
Yep.
Perfect. Okay, grand. So again, thank you very much, Peter, for your presentation. And I'll now just come on with a few slides just to delve into what other technologies are in the spatial biology arena. I know Peter has touched on these, and just compare them to QF-Pro. So I'm sure we all know spatial biology is the study of biology and pathophysiology, with a real focus on where are these events happening spatially within a sample. And it's an area that's come to real prominence in the last few years of research. And I think there's a good understanding that not just looking at what species are in your sample, but where they are, and from our point of view, what they are doing, is critical for driving research. So the kind of technologies that are currently operating in fixed tissue samples we'll focus on in spatial biology are those of immunohistochemistry, immunofluorescence, imaging mass cytometry. As Peter touched on, we have the proximity ligation assay, and then we have QF-Pro. So just to briefly go into some of them, immunohistochemistry and immunofluorescence, I think we're all very familiar with. And I think there's been a real drive in recent years to really increase the plex of what one is looking at in a sample with these technologies. And we now have platforms that are able to read out very high plex what species, what targets, what proteins, what biomarkers are present in a sample. And with this new lean on spatial awareness, we know where they are in a sample. However, these technologies, they're always going to be limited by how specific is your antibody against your target, as Peter touched on. And the whole reason we really created this QF-Pro technology was because if we have an antibody against a phosphorylated residue of our favorite target, let's say protein kinase B, we can't guarantee that it's not tagging other phosphorylated residues of other similar kinases. So these are good technologies to give you an idea of what is in your sample, and with a focus on immuno-oncology, they're valuable for showing, okay, what immune checkpoints are present in my sample, and that can be really useful for research and translating that through to clinical applications. But really, we still need that layer of functional spatial biology. And this is where QF-Pro comes in. We need to be able to report on what are those targets doing and will they respond to a therapy, for example. So what we did is we identified the other key player in the spatial biology arena, which are proximity ligation assays. Like QF-Pro, they are two-site assays that target two residues. That may be the residues of two interacting proteins or a protein and its subsequent post-translational modification. And this will use a ligation technology that when the two probes are within tens of nanometers, that you'll get a positive signal. And as you can see on this top right-hand image, you get a little red dot for where an interaction or activation event has occurred. Now, however, these assays work at 40, 50, sometimes even 60 nanometers. So whilst these technologies and proximity ligation assays are able to try and report on what's functionally happened to a target, they lack the real spatial resolution to confirm a real interaction state. They're just giving you proximity, as the name implies. So as Peter said, QF-Pro is the only technology out there which quantifies protein functionality rather than expression or proximity in a spatial manner. So as a little recap, I think Peter has explained it perfectly, but QF-Pro is a two-site assay. It's an immunofluorescence styled assay. So we have two primary antibodies against our targets. They are detected with our probes that have the donor and acceptor FRET chromophores. And critically, unlike previous FRET technologies, we have an amplification of the acceptor chromophore. So that is key in unlocking the real potential of FRET to give it the ability to make these quantifications of protein functions. So what this talk is now really going to delve into is really how does QF-Pro compare to that other two-site spatial biology technology, which is the proximity ligation assay. And really this came about as lots of research partnerships that we've undertaken and some client-based work we're doing actually has involved people that have tried the proximity ligation assay and really failed to translate it and get it over the line and give real, reliable, robust results. So we thought from HAWK Biosystems what we would do is just undertake a short comparative study, just benchmarking the technical abilities of both technologies, and then just touching upon how they may translate clinically. So within this study that we undertook, we have three phases. We started with a technical benchmarking to compare really the precision and sensitivity of both these technologies. And in this, we used a cell-based model with SKBR3 breast cancer cells, and we were looking at the activation state or the phosphorylation state of protein kinase B, AKT. Then, as Peter mentioned in his talk, we at HAWK have quite a good focus on immuno-oncology. So in phase 2, we did a technical benchmarking again in cells, but looking at the ability of both technologies to map and quantify protein-protein interactions in the immuno-oncology arena with a focus on PD-1 and PD-L1. Then in phase 3, we carried essentially phase 2, we carried it into tissue samples and compared the performance of both platforms in tissue samples and to identify whether the PLA approach could yield clinically significant data, such as the data that was yielded and generated by QF-Pro in the results that Peter shared. So, like I said, firstly in phase 1, we started with a technological benchmarking to look at sensitivity and response to experimental conditions. So on the left-hand side, we have the QF-Pro representative images. These are just some example images from the study. And on the right-hand side, we have those of the proximity ligation assay. And what we can see is on the top, we have basal or non-activated SKBR3 cells. Here, protein kinase B is just ticking over, keeping the cells alive, dividing and growing. And on the bottom, we have cells that have been stimulated for 10 minutes with epidermal growth factor. This is a stimulant known to cause an increase of activation of the AKT signaling pathway. So what we can see on the left-hand side of the QF-Pro images is a very easy to interpret and visual change in color from blue down to yellow, green, and orange. And on this color scale, this just identifies that, yep, EGF has caused quite a prominent increase of activation state of protein kinase B. And what we can really know is on the orange pixels around the plasma membrane, these indicate areas of increased activation state, and that's actually where AKT undergoes its most prominent activation kinetics. So here you can see QF-Pro is giving you a very easy and very visual representation of what is your protein functionally doing in a sample. On the other side, the proximity ligation assays, we have DAPI staining to identify the nucleus of cells, and then every red dot is essentially indicating an area where AKT is active, or activated, sorry. Now, crucially, with the antibodies being able to be up to 60, 70 nanometers apart and still yield a positive signal, this can generate and lead to some false positives in this application. And moreover, and what I'll touch upon in the next slide, is that behind every single pixel of a QF-Pro image, we have an amount of energy transferred from the donor chromophore to the acceptor chromophore. And based on that, we can work out how far away those chromophores are, and therefore have a good idea of how far away two interacting proteins are. So we've got a real amount of quantitation in this assay. And this is what gives a heightened ability to determine even very minute changes in experimental conditions. So still under phase 1, this is still the SKBR3 breast cancer cells, still looking at the activation state of protein kinase B, its phosphorylated threonine 308 residue. And what we did is did a time course of EGF stimulation. And we can see that when stimulating 1, 2, 5, or 10 minutes with EGF, we see a very prominent time response curve to EGF stimulation, when QF-Pro can really determine minute but distinct and statistically relevant changes and increases in activation state of AKT. However, the lack of quantitation that's associated with proximity ligation assays is just a very binary system giving you a dot if there is what it deems to be an activated protein. That lack of quantitation really means that it's unable to determine any significant differences between experimental conditions in this example. Like I said, we then moved on and thought once we have a good history of protein post-translational modifications, we have a very strong interest at HAWK in immuno-oncology and protein-protein interactions. So again, in a cell model, and this was a cell co-culture model between CHO-K1 and JERCAT cells, we again looked at the interaction state of PD-1 and PD-L1. And what we did was we did this in the presence of a blocking antibody. It's not nivolumab pembrolizumab. This is an experimental blocking antibody, but its mechanism of action is identical. It really just disrupts the interaction of PD-1 and PD-L1. And again, with this quantitation is the key word I keep repeating. QF-Pro is able to determine how the PD-1, PD-L1 complex is disrupted and inhibited with various concentrations of this experimental blocking antibody. Now, again, that lack of quantitation in PLA and just that dot counting of how many red dots are in my sample doesn't give enough usable data to determine experimental differences, and here failed to determine whether PD-1 and PD-L1 had indeed, whether that complex had been disrupted by this drug or not. And again, that the lack of quantitation and that working distance of tens and tens of nanometers, that's to say that proximity, and as Peter, I think, said, that's two proteins that just happen to be in the same neighborhood of a cell or even in different cells. It's really not giving you usable and translational data. So the last phase of this little study that we did was to then look at PD-1 and PD-L1 interaction states in tissue samples. And these are some examples from the non-small cell lung cancer samples that we analysed and published last year in the Journal of Clinical Oncology. And I believe that's the data set that Peter shared with us. And fortunately for a small group of these patients, I think it was around 19 patients, we had spare tissue left over, so we could actually really compare QF-Pro and that published data set. On those 19 patients, we had enough tissue to replicate the analysis with proximity ligation assay, looking at PD-1 and PD-L1 interaction states. And again, from an imaging point of view, On the QF-Pro side, you've got that very visual and easy to see change in color scale, with the pixels represented in orange and yellow, giving you the areas of very prominent PD-1 and PD-L1 interaction. Whereas on the PLA images, where we again have the nucleus stained with DAPI, and here we're looking at red dots, it's very difficult to determine a significant difference between the two conditions. And this is a patient with a low interaction state or a high interaction state of PD-1 and PD-L1. It was very difficult to really see a difference visually in that data based on the images. And moreover, and something I should have mentioned in the first set of images, is that QF-Pro, it works at a sub-pixel resolution. So we're looking at single nanometer distances between 1 to 10 nanometers. That's the area that FRET and QF-Pro is working at. So that means, despite the images, of course, being subject to the diffraction limit of light microscopy, behind every pixel, we're looking at nanometer sub-pixel events and quantifying those. Whereas proximity ligation assay images, which are very, very similar to immunofluorescence styled images, it's very difficult you're really limited by that diffraction limit of light microscopy. And here, for example, if I just put a laser pointer on, here we can see a larger dot, which here would be shown that PD-1 and PD-L1 are in proximity. It's very difficult to tell in that style of image whether that's two or more interaction events that are very close to each other, and hence that's generated a larger red dot, or indeed whether there was just more, whether there was more ligation in this assay, and that was just a stronger positive signal reporting on just one proximity event. So again, the images are, we feel, far more spatial and informative in QF-Pro. And again, it's that quantitation that's key. So for us to be able to make measurements of interaction states across every pixel of an image, we're able to generate data sets that are statistically significant. So as a disclaimer, we of course know this was just 19 patients from where we had spare tissue taken from that publication. We know that is a very low N number, but actually from this subset of patients, and despite them coming from different clinical backgrounds and different institutions in that trial, the QF-Pro, with its still cutoff of high FRET and low FRET, was still predictive of response to treatment, even in that random 19 patient subset cohort. However, the proximity ligation assay, looking at the proximity of those proteins and counting the number of red dots or the number of interactions per cell, there's not enough quantitation and meaningful data there. So it wasn't able to give anything that correlated to patient outcome or response to treatment. So within this small comparative study between QF-Pro and proximity ligation assays, we confirmed that it's the real high dynamic range and precision of QF-Pro. And by high dynamic range, we really mean that we are determining at a single nanometer distance what the difference is between the interaction or activation state of proteins. And we're able to discern very, very small experimental biological differences in response to experimental conditions. Again, proximity ligation assay has taken a big step forward away from immunofluorescence and immunohistochemistry assays, whereby it is understanding that, yeah, antibodies aren't perfect, and therefore one-site assays, which are just looking at the expression of a target, they are always going to be subject to the specificity issues surrounding monoclonal antibodies, whereas QF-Pro and PLA both take a step towards solving that with a two-site approach. It's the ability of QF-Pro to really limit those interactions to 1 to 10 nanometers that really eliminates false positive results. And lastly, whilst PLA and proximity ligation assays don't need very specific hardware, they can be run on immunofluorescence microscopes. The data analysis that was associated it was somewhat laborious in the sense that one had to create algorithms to count the number of cells, then the number of dots, discern what was a dot from a speck or a noise on the image. And on the contrary, QF-Pro being more spectrometry based is automatically giving calculations and values per image. So the processing and the analysis of the QF-Pro data sets is very streamlined and very simple. And again, and to summarize, with all these technologies, QF-Pro is the only one in the immuno-oncology domain that's really predictive and clinically powerful in the domain of immuno-oncology. And due to a lack of analytical robustness almost, proximity ligation assay wasn't able to show any clinical value in that comparative study. And I think this is just another comparison that I think I've already said. So yeah, I hope that that was an informative comparison between some other spatial technologies that exist, particularly, of course, the proximity ligation assay. Well, I'd like to thank you again, Peter, for your time and such a useful and interesting presentation. And thank you, everybody, so much for your time and interest. This has been, one thing to note, this has been recorded, so please, if anyone would like to share this with any colleagues, etc., as well, please let us know, and that's something we'd be happy to help you with, and happy to get this presentation to as many people offline as possible. So with that, thank you everybody very much, and I'll end the webinar here.
Thank you very much everyone for attending the first HAWK Biosystems webinar. After finishing the webinar, we received some questions, so we thought we'd record a short video just to go through the most frequently asked questions. Now, the first was to do with the workflow of QF-Pro and how compatible it is with current laboratory protocols. So QF-Pro itself is styled as a two-site immunofluorescence assay. Once a user has selected their primary antibodies against their targets, the QF-Pro reagent kits contain everything needed to do the labeling. In terms of samples, QF-Pro uses fixed cell samples or routine formalin-fixed paraffin-embedded tissue samples. So QF-Pro is completely compatible with current translational research and histopathology laboratory workflows. So the second question we received was to do with selecting primary antibodies that are compatible with QF-Pro. So the main requirement is that one antibody is raised in mouse and one antibody is raised in rabbit, and this is for the compatibility with our QF-Pro secondary reagents. Otherwise, we recommend that primary antibodies are monoclonal and have been verified for use in immunofluorescence and immunohistochemistry. Now, the third question we received was to do with the multiplexing of QF-Pro. So, at the moment, each QF-Pro readout will give you the expression of the donor species, so as an example PD1, the expression of the acceptor species, so for example PDL1, the functional interaction state between the two, and if a user wanted to add a third biomarker for expression, for example CD3, as long as this uses a fluorophore in this 635 nanometer wavelength, then that's absolutely fine. Moreover, towards the end of this year, we will be releasing our new QF-Pro reagent kits. Now, these kits will be multiplexable, and it will allow users to look at the functional states of 4 or more different biomarkers within their samples. A fourth question we received was, aside from protein-protein interactions and protein post-translational modifications, can QF-Pro look at protein-DNA interactions? And the answer here is yes, it can. Another question we received was to do with the hardware, and it came from a couple of researchers that had used fluorescence lifetime imaging microscopes in the past, and it was to ask, what environmental conditions can Violet be used with? So Violet is light sealed and therefore does not need a dark room. It is liquid cooled, so does not need any special air conditioning or temperature requirements, and Violet doesn't need to be placed on an optical table, so Violet can be placed anywhere that's convenient in your laboratory. And lastly, we received a question about sending samples to HAWK Biosystems. So instead of doing the labeling and analysis in your own laboratories, it's where the samples can be sent to us. So we have proof of concepts in place where we help users create new QF-Pro assays against new targets. And we also have services where we do the labeling, analysis and acquisition of QF-Pro on your samples that you send to our facilities here in Spain. And then we deliver a report with the results directly to you. So please, if there are any further questions, do get in contact with us directly or via a distributor in your territory. And we look forward to seeing you at the next HAWK Biosystems webinar. Thank you.